So I have kept the convention of this meeting alive by a last-minute change in the title of my talk. Actually, it so happened that when I made the registration, I put a rather generic title, and then I just forgot to update it later. Anyway, so this is the title of the talk. So what I'll talk about today is this little piece of work that I have done with an undergrad student at ICTM, which we basically started out as more like an exploratory project. So it wasn't really sure where it is going to lead or whether it is going to was something interesting. But because it was an undergrad project, of course, one could take some risk in that. And it turns out that we did get some interesting things, but as you will see that there are actually no hard conclusions. Many of our results are still tentative and preliminary, but I feel that there is some sufficiently interesting stuff in there that it is okay to give a 10-minute talk on it. And after Aldrich moved on to Brandeis as a graduate student, the work has been taken over by my PhD student, Arya. So what is this talk going to be about? Right, so run and tumble walk has been sort of one of the effective themes of this conference. I'm sure not by design, but we have had so many talks on it. And as you know, run and tumble walk is the sort of mechanism employed by bacteria like E. coli to move around, and it's the sort of modification of this walk by in the presence of external gradients that helps it moves, it find food and so on. While I'm talking, you can actually watch this guy moving. This is actually not a bacterium. It is actually a much larger eukaryotic cell. It's a neutrophil in our blood, a white blood cell. And here you can see it actually chasing a bacterium and finally engulfing it and eating it up, right? Now, of course, a eukaryotic cell also performs chemotaxis, which is, of course, a very generic mechanism employed by many different kinds of cells to move up chemical gradients in search of food away from harmful materials and so on. But the way that these guys detect gradients is quite different because they have sufficient spatial extent that they can measure concentration differences across the length of their body. So the question is, what are the sort of interesting aspects of eukaryotic cell motion, especially when you have multiple cells involved? So that will be roughly the subject of my talk here. Now, the details of the sensing. So you have this cell here, which is visualized as a sphere, and your receptor proteins sort of spread out all over their surface, and you have this external signal, the ligands. And the ligand binds the receptors, activate them, sets off a chain of reactions. And finally, what happens is the cell itself gets polarized, usually accompanied by shape changes. And the polarization basically refers to localization of specific signaling components asymmetrically over the interior of the cell. And that sort of gives the cell a certain polarity, which one can sort of approximately visualize as a vectorial quantity that will essentially guide the cell in its motion. So here actually this picture shows that there is more fluorescence on one side of these cells compared to the other side, and that is because in between here there is a source of cyclic AMP, which is a chemoattractant for these dictyostelium cells. So next question is like, if you have a situation where all these cells, so these are like little compasses all moving together, and of course it is very tempting to think of what will happen when these compasses or vectors tend to interact with each other. Of course they do interact. Collective motion of chemotaxing eukaryotic cells is of course a very well-studied field, experimentally as well as theoretically. There has been a large number of fantastic theoretical papers in the area, also sort of intruding into active matter research. Now, only problem is that the interaction between these kind of objects, a cell being what it is so complex that we can't really sort of write it as a Hamiltonian or anything like that. So one has to sort of study different types of experiments and sort of try to deduce the nature of these interactions. So I'm actually not going to go into all those possible experiments that one could possibly look at, but I'll just focus on one particular type of interaction, which is extremely common in chemotaxing eukaryotic cells, and that is called by the generic name of contact inhibition of locomotion. And you look at this sort of these two pictures side by side, what you see here is that there is a gradient that is enforced here, and here you have two cells. One is the leader, one is the follower. And here you have a situation where actually one cell actually hits against the other. And there are two possible outcomes that are reported in this particular experiment. So in one case, you see that actually the cells, the trailing cell effectively becomes a follower of the leading cell, and both the cells move up the gradient in a disciplined manner. But it could also happen with a certain probability that the trailer cell actually completely reverses its polarity and at least temporarily moves temporarily moves down the gradient. Okay, and the interesting thing is that the relative probability of these two outcomes was found to depend on the mean concentration of the attractant molecules in the surroundings. Okay, so this is what actually sort of caught our attention, and we sort of started to ask ourselves, can we sort of think of a simple physical mechanism based on the dynamics of the diffusing concentration field that can account for both these phenomena and possibly also quantitatively produce the relative probabilities of these events. Of course, we haven't been completely successful. We made some progress though. So I would sort of now quickly sort of tell you about the very simple model that we started with, and that's think of each of these cells as an absorbing sphere, and the chemoattracting concentration outside in stationary state satisfying Laplace's equation as it does. And think of all these ligand molecules that are coming close to the cell are just being sucked in by the cell. And then the question is, if you have an externally imposed concentration with a gradient present, what is going to happen? Now, this problem can be very nicely mapped to a simple electrostatic problem where your cell, the sphere, actually becomes a grounded conductor, and we have an externally applied electric field, and you are looking at what is the asymmetry in the induced charge distribution on the surface of the spheres. And the answer is well known. One can find it in any sort of undergrad electrostatic textbook. And you can also calculate the dipole moment for this problem, and that of course is the analog, or that rather what we take to be the analog of the polarity vector of the cell. And then of course what we wanted to see was that of course there are no surprises here. It is proportional to the externally applied gradient or field as the problem it is. Next, what we wanted to see was that what would happen, of course, if you bring two such objects close together, and then you have a situation where you have Laplace's equation outside the field, and then you have a boundary condition satisfied on both these spheres, so needs to be satisfied. Well, electrostatics again guides us the way. You can write a general solution for the concentration field as being as a sum of the imposed external field, to which we add the contributions coming from the induced charges on both these cells or spheres, conductors, as the case may be. Now, of course, we don't know these charge densities beforehand. We need to find them. Well, we have the simple boundary condition, the Dirichlet boundary condition on the surface of the spheres absorbing, so which we impose, and then we expand the charge densities in spherical harmonics with a distance dependent coefficient. One can solve it perturbatively, inverse power of the separation between the spheres. Well, you do get nice results. Only problem is that the results are sort of perturbative, so we can't really apply them when the cells come too close to each other. But whatever we got is still sort of instructive. In the absence of a gradient, of course, the dipole moments point away from each other, because that is what they need to do. With a gradient present, well, there are actually very interesting effects. So you have sort of the repulsion part actually adding to the alignment part, and depending on the relative distance between the spheres, the cell may like to align along with the gradient, or they may actually like to orient away from each other. We just wanted to check it in simulations. So we did some simple Brownian simulations, and this is actually just a pictorial depiction of what we sort of expect when you have multiple spheres in the problem. They're sufficiently far away, you expect alignment because the gradient dominates. Sufficiently close together, the repulsion dominates, the cell point away from each other. In some way, this is qualitatively similar to what was observed in those experiments. Except that, of course, we don't have any dynamics in the problem. We just sort of have static spheres at specific locations. Well, we did check it in simulations. Unfortunately, for some sort of circumstantial reasons, we couldn't really sort of simulate really large cells, like size of eukaryotic cell. This is quite small, but we did see that as we sort of in V gradients, actually, we don't see much of a polarization here. This is actually cos of theta, which is the component of the dipole moment parallel to the externally applied field. Well, small gradients actually, irrespective of the separation that you put the spheres from each other, we don't really see much of polarization. I'm almost done. We go to slightly sort of stronger gradients, and we do see that the distribution actually starts developing peaks away from the origin. And the leader cell is, of course, polarized always parallel to the gradient, while the trailing cell now, when you sort of slowly increase the gradient, it starts to polarize away from the gradient. Now, that is just what we wanted to see. Well, unfortunately, the only problem is that the size of the cell was so small, and I should be candid enough to admit this, that the noise or the fluctuations is actually quite large. So actually one shouldn't sort of treat these conclusions as decisive. So we are now in the process of repeating the simulations for much larger cells, as well as trying to do some sort of more detailed theoretical analysis with extrinsic and later also intrinsic cellular level noise built into the problem. Okay, so you can sort of read up my conclusions here, and that's where I'll stop. Thank you very much.
Questions?
You're talking about the charge density distribution. Okay. Yeah. Right. 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 Absolutely. Yeah. Yeah. Yes. 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 Of course, we have only looked at spheres here. Is it? Right. 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 Okay. Okay. Yeah.
I mean, so it looks like the cells have some in their orientation, some interaction is generated.
Yes.
But what about
That
like
is of course well known.
position? Yeah, what about like just positional in space? I mean, is there some effective?
There could well be, because one cell could be signaling to other cells by secreting signaling molecules, specific signaling molecules, but that is not what we wanted to look at here. So there are actual specific biochemical.
No, in terms of like competition for like resources, or I mean, because they're consuming resources, right? I mean, so that doesn't generate any.
Well, the orientational sort of the repulsion sort of to some extent takes care of that. In fact, that is one of the hypotheses, that the reason why the cell would find an advantage is that they would actually tend to move away from each other, so that each of them would be sort of better equipped to use up the available resource. In fact, sort of proliferation of cancer cells, for this the contact induced repulsion is sort of thought to be a very useful mechanism. They do tend to move away from each other.
Okay, if there are no other questions, let's thank Manoj.